processes that you would get off-target effects. And indeed, the Green Revolution varieties are thought to have less nitrogen use efficiency, for example. So breeders are trying to breed in better fixes on that side effect of manipulating GA levels with sort of a broad manipulation of GA levels. So that indicates to me that we should really try and understand these things in a finer level. In particular, even though the decision-making for a hormone to whether to accumulate or to respond to a given hormone is largely cellular, because other parts of the plant are also making and potentially sending hormone signals that or catabolizing them, that a cell can also be integrating systemic information by processing with hormone accumulations and distributions. All this is to say that we should probably, I would argue that to understand information flow through hormones, we really need cellular all this is to say that we should probably, I would argue that to understand information flow through hormones, we really need cellular resolution over time. So we need to understand the cellular hormodynamics of cells in a tissue in a living organism, or even across the whole organism. So that's not that easy. So I've actually been interested in tracking cellular hormodynamics for a long time, like I said. So in my undergraduate, I was working on signaling reporters for auxin. In my PhD, I did HPLC and transcriptional response reporters for salicylic acid. At the end of my PhD, I realized, this is not the best way to do this. So Wolf Fromer at Carnegie Institution was developing these lovely sensors, FRET biosensors for metabolites. And I thought that's the way to do it. So FRET biosensors can be directly binding to a ligand. They can be ratiometric so that they are, which is really great for quantification. They're genetically encoded, which means you can target them to many cells and also different organisms. And they are high resolution. So you have very high spatial temporal resolution. And if you target the direct sensors to subcellular compartments, you could even get subcellular information on hormone dynamics. So that's a really good bit of information to start the rest of those investigations from. So I sought out to engineer biosensors for all the plant hormones, actually. And the idea was to make a library of ligand sensory domains that would interact with the hormones, and a library of fluorescent protein FRET pairs. In this case, we always use CFP and YFP variants. And the way these things work is once you get one fusion protein, that fusion protein can change its conformation based on the presence of the ligand. You can get more yellow emission or less yellow emission after ligand binding in relation to the cyan emission when you're only exciting the cyan protein. So you get an emission ratio, and that's proportional to the ligand concentration. So the idea was to make hormone binding proteins, hormone binding sensors. And the first sensor I was able to engineer was for abscisic acid, or ABA. And I called this Abacus, or ABA concentration uptake sensor 1. And around the same time, Rainer Watt in Julian Schroeder's lab was able to engineer abalians. And so both sensors actually worked in very similar ways with an ABA binding domain, fluorescent proteins variants of CFP and YFP. And they were giving FRET ratio changes, emission ratio changes in response to ABA. Now, at the time, we had really good tools now for measuring ABA in vivo. It was a very exciting time. But already I knew when I started my group in Sainsbury Lab in Cambridge that we could do better. So I sought out to engineer Abacus 1 with the best properties of Abacus, which was a high emission ratio change in response to ABA, so a good signal to noise ratio in vivo, and the best properties of abalians, which were the higher sensitivity to ABA. And so it's been many years, and I won't tell you all the details, or very many details at all, other than to say for the affinity increases, we engineered the binding pocket around ABA, as well as other residues in the protein to get a higher affinity, whilst maintaining that ratio change that I talked about, the emission ratio change. And so this is our random walk through the engineering forest. And what we see is that eventually we did arrive at high affinity and high ratio change sensors that we were excited about, called next generation Abacus 2 sensors. And this is a emission ratio in response to different concentrations of ABA in vitro. And this is showing you that the new sensors are responding to lower concentrations of ABA with substantial ratio changes. So these are going to be ideally, hopefully, better in vivo when expressed in plants. So what we did was we took these fusion proteins, the genes for these fusion proteins, expressed them in Arabidopsis. We also have other plants now, but this study is all in Arabidopsis. And this is the shoot system of a Arabidopsis seedling. And this is what we get out. So this is a false colored rainbow image where reds and yellows are representative of high ABA levels and blues and purples, lower ABA levels. You're seeing dots because we like to send this fusion protein to the nuclei of plant cells. That helps us with imaging, higher quality imaging, and also it helps us discriminate neighboring cell types in analysis afterwards. So we can see neighboring cells, which ones have more ABA, which ones have less, and also what happens over time. So already you can probably intuit that we can do a Z-stack in a confocal microscope and discriminate which cell types are having, what ratios the different cell types are having, which is representative of how much ABA they have. And some cells indeed do have more ABA. For example, the vascular strands tend to have higher ratios and higher levels of ABA, and that is thought to be a major site of synthesis in leaves. So we can do lots of different tissues and lots of information about ABA dynamics. And what I'll tell you about today is one of the stories Jim Rowe, in the lead postdoc on the project, has developed around systemic ABA signaling. So this idea that some parts of the plant can talk to others via ABA. Now, you might already be thinking, I've heard this story before. And that's because in the textbooks, ABA is thought to be discussed as a mobile signal in the xylem that communicates from roots to shoots. So when you have soil drying, you have an accumulation of ABA that will move up in the transpiration stream to the leaves, where it eventually accumulate in guard cells, close them, and thereby conserve water. So there's a way for roots to communicate with shoots. Now, this is an old model. Subsequently, several other signals have been proposed to communicate soil drying from root to shoot, including sulfate, Ple25 peptides, and even hydraulics. So the xylem column can have its own pressure signals that maybe can affect leaf synthesis of ABA in those vascular cells or even in the guard cells. So when we first had the sensors, so both I observed this and Reiner observed this, that we could actually see ABA not moving exogenous ABA when we apply it to plants. Rather than moving root to shoot, we could actually see better evidence of ABA moving from shoots to roots. So if you apply ABA to the leaves, you see it in the roots. So Jim was able to recapitulate this experiment with Abacus 2, and this is a different lookup table. So basically yellows are high ABA and blues or purples are lower ABA. This is more friendly for anyone who's colorblind in the audience. So you can see ABA accumulating in certain cells over time, of course, of an hour or two. You can see higher ratios, higher levels of ABA in the roots when the ABA was applied to the leaves, suggesting that indeed exogenous ABA can move from shoot to root. But what would this mean in endogenous ABA? Is the plant using this potential movement of ABA from shoot to root in any meaningful way? And we didn't know the answer to that, but we did have a hint. And that is from another study that was showing that ABA is actually leaf autonomous ABA, let's say, can be important for humidity responses of leaves. So in this experiment, an ABA biosynthesis mutant ABA3 was much more wilty and dry air. So this is 3 hours of dry air, suggesting that, and this is a detached leaf, of course, so the root's not involved. So yes, ABA probably can be synthesized in the leaves. So we used Abacus to detect what's going on in the leaves with the dry air treatment. And so Jim was able to show that as we go from high humidity to low humidity, we can see ABA levels increasing in certain cells of actually several types of cells in the leaves. So indeed, we think ABA is accumulating. And this response happens over a period of hours. And he also looked at the roots. So this is a root system that is hydrated, it's well watered, but the leaves of this plant are experiencing low humidity. And what he found was that indeed there was ABA accumulating in the root system, particularly in lateral root primordia, in the vasculature, and in the primary root. near the root tip when a leaf was experiencing a humidity drop. So if I just freeze this movie for different time points, you can see that after 0 hours of the humidity stress to increasing time, you can see an accumulation of ABA, particularly in the elongation zone and in the root tip, for when the leaves were exposed to a humidity drop. So what could this mean? Is there any functional information in this ABA? And indeed, when we have an ABA2 biosynthetic mutant, Jim has found that while this mutant grows roots pretty normally under mock conditions, so well-watered roots and humid shoots, when the shoots are experiencing a humidity stress, you see a much reduced root system for the ABA biosynthetic mutant, suggesting that ABA is important somewhere in the plant during humidity stress to protect root growth. So oftentimes ABA is thought of as a growth inhibition signal, but indeed sometimes it is a growth promotion signal, or growth, in this case, growth protection signal for roots. So we don't yet know what the signals are that would halt growth in ABA2 mutants, so why these roots are not growing. We just know that ABA is acting to allow wild-type plants to grow well under humidity stress. But we could ask the question, where is the ABA acting to promote growth or protect growth? So which of these cells is important in the plant? So what Jim decided to do was take a signaling mutant, SNRT22, SNRT23 double mutant, and complement it with a specific promoter. So this mutant is indeed sensitive to humidity stress in the leaves. The roots are growing slowly, and the complementation line in the root tips, this RCS1 promoter drives expression in the root tips, behaves very nicely, grows very normally. So indeed, the root tip accumulation of root tip ABA signaling is good to complement the phenotype. So we do need ABA signaling in the root tip. Interestingly, this is the site of phloem unloading in the root tip, and it's the site of ABA increase, suggesting that maybe there's a phloem mobile ABA signal, as we have observed for exogenous ABA. And indeed, when we induce catabolism by overexpressing a catabolic enzyme, CYP77A3, in the phloem under the control of a SUC2 promoter, we do see hypersensitivity to humidity stress in the leaves and roots. So roots are slower to grow when the leaves are under low humidity in the induction of this catabolic enzyme in the phloem. So we do think indeed phloem is important. So we found that with Abacus2 biosensing, we found a lot of different patterns. This is an entire seedling that we're showing you here. Guard cells, epidermal cells, root cells. This story will come out. The study will come out very soon to be able to examine all the different measurements Jim was able to make. And today I've told you that we are quite interested in this, the idea that endogenous phloem ABA is a root growth signal during leaf humidity drop. And so we think we've identified a function for this ABA that we thought was moving from shoots to roots. So there is a function for ABA in communicating from shoots to roots that we think we can continue exploring in the future. And we think this is, one thing I would like to mention first is that this is for everyone, right? So Abacus2 is a tool. And in fact, Poonam Mehra and Malcolm Bennett's lab has already used Abacus. Her recent science paper studying root branching affected by ABA and auxin dynamics. So really, this is hopefully everyone else can start using Abacus2 and our other hormone biosensing tools for whatever their favorite biological question is. And going back to ours, we think this idea that the leaves are experiencing humidity stress could affect the roots is really important, because in the context of irrigation agriculture, where you do have hydrated root systems, but the leaves may be experiencing, or the shoot system may be experiencing, very dry air. And this can be a stress sometimes. And we think that maybe we can help plants cope better with the stress by understanding the way ABA can function in this signaling. And this will be increasingly important with climate change, as very dry air spells are expected to increase. And we think that, of course, we would like to think whole plant and cellular and subcellular scale on this, to say what are the integration patterns, what's the information processing, why are they hormones? Okay, so I've told you about our work on Abacus2, both engineering and one of our biological stories about shoot to root signaling. And now I'd like to tell you a little bit about our work on GPS-1 with gibberellin perception sensor 1, which was a sensor I started engineering in Wolf's lab, and continued to develop in my group in the Sainsbury lab. And Annalisa Rizza has been doing this, a lot of imaging with GA in the root system of Arabidopsis, and particularly trying to understand what is the relationship between this cell gradient and cell length in the roots and gibberellins. In particular, because when we started using GPS-1 in Arabidopsis roots, we saw very quickly that there was a accumulation gradient of GA in the roots. So we see that there's a correlation with cell length in Arabidopsis roots, and this was actually a really big surprise because there was already a study that said that GA would actually be anti-correlated with length, in particular because of this expression of this biosynthetic enzyme GA20 oxidase 1 was more in the root elongation zone. Can I ask what my time is? I've lost my timer.
Hello, Jones. You still have 3 or 4 minutes there.
Okay, thank you. Okay, so I'll move quickly here. So we actually revised the mathematical model that Leah Band and Marcus Owen had originally developed, which said that GA was anti-correlated with length. And we found that a revised model with GA increasing in the elongation zone was actually better representative of the findings of our biosensor. And so we then decided to look at which biosynthetic steps were likely rate limiting. And we found actually some surprises. So early biosynthetic steps, we could probe when we add the precursor, GA12, were not very effective at changing GA levels in Arabidopsis roots. But when we overexpressed what was thought to be rate limiting GA20 oxidase 1, we also did not see a strong effect. When we overexpressed the last biosynthetic step, we saw a big dramatic effect in the elongation zone. And interestingly, there is one enzyme, GA3 oxidase 2, that better represents this GA pattern in roots. And we think that would be a better candidate for example, integrating environmental information into gibberellin patterns in root growth. So there was still more to the story. One thing we found was that both biosynthetic enzymes, 20 oxidase and 3-os, the last 2 biosynthetic steps, are both rate limiting in the root tip. And when we add all 3 together, we can see that there's even more accumulation in the elongation zone, but there's a really interesting depletion still, even though we're providing the immediate precursor in both of the last 2 biosynthetic steps in the elongation zone. And I'm running short on time, but I will tell you that we have found that this particular depletion can be manipulated, and we think it's due to the fact that these enzymes, both 20 oxidase and 3 oxidase, are both 2 oxyglutarate dependent dioxygenases. And they're both using 2 oxyglutarate as a co-substrate. So we think that concentrations of oxyglutarate are important for gibberellin biosynthesis. This was a pretty big idea for us because oxyglutarate is right at the heart of carbon and nitrogen metabolism in plants. It's present in your body, of course, and regulates 2 oxyglutarate dependent dioxygenases in humans and very important biosynthetic pathways. Could this be actually a way for primary metabolite to regulate these important enzymes, which had never been shown in vivo? And so Annalisa has been able to show that oxyglutarate does indeed induce GA accumulation in roots, even in the even in the wild type context. So we think it is indeed rate limiting in some contexts for gibberellin biosynthesis. We've been able to show with some control biosensors and some biosynthetic mutants that it's not an artifact of imaging or something else going on with the sensor. We've been able to show with the signaling reporter with DELA GFP that indeed this GA accumulation does seem to signal and act to degrade DELAs. And even if we provide an inhibitor of the next enzyme in the pathway in the Krebs cycle, we will accumulate oxyglutarate, and we do see an increase in GA in that context. So we think, in just quick summary, that with engineering GPS-1 and deploying it, we've overturned and corrected multi-scale mathematical models. And so this is already published. We've identified cell-resolved rate-limiting biosynthetic steps in GA biosynthesis. So very important, it's not one enzyme that is necessarily rate-limiting for a pathway. You really have to think cellular scale and over time for this. So doing the biochemistry in vivo was very powerful. And we think we've discovered a novel mode of regulation of GA biosynthesis, which could potentially impact duo-ODD enzymes in all other systems as well. And that provides a direct link between energy metabolism and a growth hormone. So to zip through some big picture stuff, we have GA and other organs, of course, GAPS imaging in other organs and other species. We didn't tell you about our functional information, where we think the GA is important. Part of that is because we're still ongoing studies in this, although we do think that, for example, in Medicago, we know that GA is important to be in some places and absent in others. And we're developing new tools to measure, to perturb hormones in vivo. And this has also been published in bioRxiv. So this is our new highlighter tool, or optogenetic gene expression tool for gene control in plants. So we're working on this. This is transient tobacco. We can do cell level manipulation of gene expression with this tool. We have the sensors in other species as well. So hopefully everyone will be able to use these sensors in their favorite species soon. And we are also developing new versions of GPS2. Auxin biosensors, Iago 1 and Iago 2 are ready to go. And Salix, our brand new sensor for salicylic acid, we're very excited about for in vivo salicylic acid sensing. So I've told you a little bit about ABA and GA, biology in vivo, and we would like to continue investigating cellular hormone dynamics across a series of environmental changes, particularly future environments that we expect to have with climate change and how they affect physiology and development. And I'd like to, of course, I've already shown the pictures of the group members that were involved, but thank all the past and current group members, our funding sources for the various projects, and of course, all of our collaborators. And I'd also like to thank you for your attention.
Okay, thanks, Dr. Jones, for sharing the interesting work on hormone biology and biosensor development. Now let's start the section of Q&A. So any question from the committee members, please?
Sorry, yeah, maybe I can start here. I just was thinking, for instance, for this threat sensors, it's really fantastic. It's really a very interesting tool, and it's going to change how we look to plants and how plants are reacting on environmental conditions. It's really going to be a big change. But I was wondering, so the whole thing works from a nuclear expression. So actually, the threat biosensor is so what is happening? The threat is occurring in the nuclei of the cells. So
yes, oh yeah.
And you in your stories you presented them as having a tool in hands to study the distribution of these hormones in plant tissue. So are we sure that we don't miss anything if because for instance, just as an example, phloem sieve elements do not have a nucleus. You see, these are the very special cell type in plants that do not have a nucleus. So
yeah, so there's no problem with targeting the sensor to the cytosol and the nucleus cytosol. That's fine, and we can look at. We have those lines as well, and they typically behave similarly. If we were particularly interested in phloem, yes, we would target the sensor in a different way. But for most of our analysis, we think the nuclei are easier to use. But there's no problem with targeting to subcellular compartments either, except for we think we might have a little challenge with low pH environments, or particularly pH dynamic environments. And so we're trying to engineer new sensors that are more resistant to changes in pH, for example. But yeah, we have cytosolic versions of the sensors. That's no problem.
Okay, so it might be that some of the ABA transport and so that is going from cell to cell that is actually never reaching the nuclei or something of these cells. I don't know. It's just I was just thinking that you might miss part of the transport route of ABA from shoot to root by only using the nuclear marker. No.
That's a really good point. And in fact, particularly for auxin, we would really like to get extracellular sensors. We think that would be a very interesting thing to look at, but we could do that for ABA as well.
So, and now having mentioned auxin, so I'm just wondering why do we need another biosensor for auxin? I mean, there must be reasons for that, but I just want to hear them from you.
Yes. So we have our affinity is higher, our sensitivity is higher than anything that's out there now for our threat biosensor for auxin. and we think we're better at capturing endogenous signaling or endogenous accumulations of auxin.
Okay, I see. Looking forward to it. Thank you.
Thank you.
There's still 1 minute left. Is any question from the committee members? Okay, if there's no question, thanks,
Yeah,
I think
Dr.
there's-
Jones.
I think there is a question somebody did not unmute, but maybe I throw in mine, just following up on Tom's. I'm wondering about this affinity thing, because I think in your optimized ABA sensor, there was written 100 N. So does that mean 100 nanomolar KD?
Yes.
And I wonder whether a sensor can be too good, because I imagine that at one point you would saturate it very easily if it is having a too high affinity, and then you maybe don't really see the dynamics anymore. And I mean, do we really have an accurate measurement of the average hormone concentrations in a cell?
So I'll take the first question. Yes, the idea is that we have multiple affinity variants for the different sensors that will capture the range of interest. So we think, for example, for the gibberellin sensor, the high affinity version is 20 nanomolar, and that's capturing a lot of interesting dynamics. There's a lot of changes that we can see. We do think there might be some interesting GA dynamics below that concentration that we might be missing, for example. And that's why we engineered, for example, the higher affinity ABA sensors. Now, the second question was more of a general question about, are we capturing the actual average? I think the sensors are basically real time. So I think over time, if you image the same cell, you might have a better idea. We do see with time courses that the cell can, we do have on rates and off rates within our root chip experiments, for example, I didn't show of rates of accumulation and depletion. So with mathematical modeling, with calculations from parameters that we calculate in vitro, we can then get an idea of what's going on in a holistic sense. That's what we did with the GA sensors with Leah in the roots. So eventually, if you want to, you can get the more quantitative analysis done, but there's a lot of parameters involved in that, so it needs some mathematical analysis.
Okay, thank you.
Okay, thank you, Dr. Jones. Yeah, now let's move. Okay, thank you, Dr.
Thank
Jones.
you.